Hello, my name's Arif Khwaja. I'm a consultant nephrologist at Sheffield Kidney Institute. I'm going to talk in this second lecture about CKD MBD and just give an overview of what we actually mean by the term CKD MBD, or chronic kidney disease mineral bone disorder. So, CKD MBD is defined by KDIGO as a systemic disorder of mineral bone metabolism due to CKD, and it's manifested by one of three different things, either by abnormalities in calcium, phosphate, PTH, or vitamin bone, abnormalities in bone, either turnover, mineralization, volume, or strength, or abnormalities in the blood vessels, or abnormalities in soft tissue calcification. This is the KDIGO classification and definition based on 2009. The 2017 guidelines haven't as yet been published at the time of recording this lecture, but I would strongly recommend you go to the CKD MBD guidelines when you start the HOC. I don't think there are going to be that many differences from the 2009 guidance as far as I can tell, although there may be some slight differences in terms of recommendations, but I think the definitions of CKD MBD are likely to stay the same. So this is just pictorially just saying what I've already said, which is that CKD MBD is characterized by a triad of laboratory abnormalities, bone abnormalities, and calcification. And what we talk about is vascular calcification. And the triad of those three different problems leads to the problems we all see in our dialysis patients, which is high risk cardiovascular disease, high risk of death, and a higher risk of fractures. So let's just talk in a little bit more detail about each of these different aspects. So let's talk about the laboratory abnormalities first, and calcium, phosphate, and PTH. So as you know, as your GFR declines, as we've already said, the parathyroid hormone here in green declines, and this starts to rise, and it rises relatively early on, even when the GFR falls to less than 80 and 70, you can see, and then as your GFR declines further, it declines more. Because high levels of parathyroid hormone maintain the serum phosphate, the serum phosphorus on the whole tends to be pretty well preserved until relatively late here in red, in this sort of pinky red here. And the serum calcium, again, because PTH mobilizes calcium from the bone, calcium is relatively well maintained in most patients, even in advanced CKD. So why do we worry about these laboratory levels? And the reason why we worry about these laboratory levels is that they are associated with a high risk of death. So if we look at parathyroid hormone, high levels of parathyroid hormone associated with high risk of death, low levels also associated with high risk of death. And if we look at all of these parameters combined, calcium in blue, phosphate in purple, and parathyroid hormone in green, you can see that once you're outside the KDOQI range, the old KDOQI range, the risk of death gradually increases at the worse your bone biochemistry becomes. And similarly, if you go the other way and the calcium is very low, the phosphate is low, or PTH is low, the risk increases. So you get this kind of U-shaped curve associated with increased risk of mortality as these laboratory abnormalities get worse. And this is important because when you attribute the risk of mortality to calcium, phosphate, and PTH, you can see when you combine all these parameters, the attributable risk and mortality risk is much higher than, say, something like anemia or inadequate dialysis. Okay, so these matter. This data seems to suggest that abnormalities in calcium, phosphate, and PTH are important in driving mortality in patients on dialysis. And this graph just tells you what you already know, for those of you who work on a dialysis unit, that managing calcium, phosphate, and PTH is really, really difficult. In this particular publication, only 30% of people achieve target PTH levels, and only 50 to 60% of people achieve target parathyroid, calcium and phosphate levels. So very, very difficult to achieve. What about the bone? What are the abnormalities we see in bone? Well, of course, the only way we can diagnose bony abnormalities is to do a bone biopsy. And KD have suggested that when we do bone biopsies, which is something that very few centres around the world do, a very, very difficult thing to do, technically difficult biopsy to interpret. What KD have said is that we classify the biopsies according to turnover, mineralisation and volume. So if the turnover is high, we call that osteoarthritis fibrosa or perhaps a hyperparathyroid related bone disease. If the turnover is low, we call that adynamic bone disease here. Then you can look at the mineralisation along this axis, and so if you've got high turnover bone disease, you may have normally mineralised bone here, or you may have abnormally mineralised bone here in blue. And if you have low bone turnover, you can have adynamic bone disease where you have normal mineralisation but low turnover, and you can have osteomalacia where you have low mineralisation and low turnover. And then the bone volume here varies according to the type of disease as well. Does any of this matter? Well, yes, fractures are more common in chronic kidney disease. As your GFR declines, you're much more likely to fracture. And patients on dialysis are much more likely to have hip fractures. These are in blue. It doesn't matter where you live around the world, this is all DOPS data. Much more likely to have hip fractures than the general population in red. And if you get a hip fracture and you're on dialysis, it's bad news, because you can see the risk of death in blue and the risk of hospitalisation in blue is much higher for the dialysis population in blue than the non-dialysis population in red here and here. What about the blood vessels? What happens to the blood vessels? Well, patients on dialysis have an increased risk of vascular calcification, as I've already mentioned in one of the previous lectures. So if you do CT scans of patients' coronary arteries, you can see in grey here, as you get older, you get a bit more coronary artery disease, and that's very common. But if you look at patients on dialysis, so this is coronary artery calcification score, you can see they're much more likely to calcify. So a 40-year-old on dialysis is still much more likely to have calcified coronary arteries than somebody who's 60 to 70 not on dialysis, and that gets worse and worse as your age increases. So dialysis patients have an increased risk of cardiovascular calcification. And does it matter? Well, yes, it does. This is probably one of the most famous slides in nephrology. It's a slide from Rob Foley looking at USRDS data, and it just goes to show that if you're young in the dialysis population, your risk of cardiovascular mortality is about the same as a 75 to 85-year-old in the general population. It's a huge relative increase in relative risk of cardiovascular mortality, although the age effect in the dialysis population isn't as pronounced as in the general population. So bad news, really. If you're on dialysis, very high risk of cardiovascular death. And in fact, if you look at certain studies, about half patients with who start dialysis already have evidence of coronary artery calcification. So this is a non-contrast CT, just showing the calcified coronary arteries, and that's progressive. So as you continue on dialysis, that calcification gets worse. A little bit about what's driving the calcification process, the pathophysiology, if you like. Well, there are inhibitors of calcification, and they're highlighted here. So things such as fetuin A, pyrophosphate, osteoprotegerin, matrix-Glar protein. Interestingly, some of these inhibitors, such as fetuin A and matrix-Glar protein, are vitamin K dependent. So if you give something like warfarin, which is obviously a vitamin K antagonist, you inhibit the activities of these inhibitors of calcification, and therefore you get increased vascular calcification. And then there are active inducers of calcification: calcium phosphate, PTH, uremic, various uremic toxins, reactive oxygen species, various inflammatory cytokines, all thought to play an important part in both vascular calcification and valvular calcification. So what about the molecular mechanisms that underlie vascular calcification? So this, I'm not going to go into this in a lot of detail, but this diagram just shows what would happen to a vascular smooth muscle cell and how it can change its phenotype to sort of calcified cell or a bony type phenotype. So you have these phosphate co-transporters, PIT1 and PIT2, and the PIT1 is the one that's more associated with vascular calcification. It induces the apoptosis of vascular smooth muscle cells and releases these calcium phosphate loaded extracellular vesicles, which promotes calcium phosphate deposition, and it also encourages, acts on various smooth muscle cell genes, so directly sort of genomic effect, encourages this sort of transdifferentiation so that the vascular smooth muscle cell phenotype becomes more like a bone phenotype, an osteoblast phenotype. PIT2 is the transporter that's thought to maybe inhibit calcification, but obviously in chronic kidney disease, PIT1 is the main driver of kind of molecular mechanisms of calcification. Okay. So if you just talk about that in a bit more detail, you've got a vascular smooth muscle cell here, you've got blood going through the middle, you've got your red blood cells there and your white blood cells in blue. And if we just look on the left-hand side, you can have passive deposition of calcification. That's thought to be relatively rare, but basically, or not the primary driver of calcification, where you get the formation of a particle known as calciprotein, which is a sort of mixture of calcium and phosphate, and initially calcium phosphate sort of complexes are scavenged by phetuin A, but that is then that process can then be overwhelmed, leading to kind of primary calciprotein particles that evolve into a sort of secondary particle. So that's one aspect of if you like, passive calcification. Then you can see that then what we see here and here is kind of phosphate particles entering the vascular smooth muscle cell through PIT1 and promoting a kind of osteoblastic phenotype, so changing the phenotype of the smooth muscle cell into a bony phenotype, so you've got a more active process there. And this here is a kind of histology of a vascular smooth muscle cell, and in red you can see the specks of vascular calcification. So, as you know, in kidney disease, the calcification mainly occurs in the media rather than the intima. So this is all referring to medial calcification. And then again, you've got these inhibitors of calcification and agonists of calcification. And so the relative balance of calcification or not will depend on the activity of these. And that's why there's a lot of interest now in vitamin K as an inhibitor of vascular calcification, and there are a number of randomized controlled trials going on at the moment looking to see the effect of vitamin K in the dialysis population, because they will increase the activity of matrix glycoprotein and phetuin A and therefore inhibit vascular calcification. And as you'd expect, if you're diabetic, here in the light blue, you're much more likely to have calcification, whether you've got kidney disease or not, than if you're non-diabetic, which is in green here. So diabetic renal disease, bad news here in the light blue, you do very badly. Whether it's coronary artery calcification or valvular calcification here, you're much more likely to have it. So unsurprisingly, diabetes is a big risk factor for vascular calcification. And as we've already shown, the worse the calcification, the higher the risk of mortality. So this is no calcification here. This is medial calcification, and this is intimal calcification. You can see the more extensive the calcification, the more likely you are to die. And again, this is data from the UK, which just shows that not only is calcification, vascular calcification associates with stiffness, and again, you're more likely to die if you're calcified when you're starting dialysis than if you're non-calcified and starting dialysis. So clearly preventing calcification, in theory, will be an important endpoint in terms of improving CKD and dialysis outcomes. What about FGF23 and the role of calcification? We talked a lot about FGF23 in the previous lectures. I think it's fair to say, if you look at this recent review, we don't know whether FGF23 from animal models increases or actually reduces the risk of calcification. There's some animal data suggests increases the risk of calcification. Other data when you give recombinant FGF23 where you have absolutely no effect or actually have an inhibitory effect on calcification. So you can see there's a variety of different effects. So we don't really know yet what the role of FGF23 is on vascular calcification, or it certainly appears to be unclear. So if we just conclude, CKD MBD refers to a spectrum of disorders caused by abnormal mineral metabolism in CKD. There's strong observational evidence demonstrating that CKD MBD is associated with significant morbidity and mortality, and it's characterized by the triad of laboratory abnormalities of calcium, phosphate, PTH, and vitamin D, abnormalities in bone, turnover, mineralization, volume, growth, and strength, and abnormalities in the blood vessels or other soft tissue with evidence of calcification. Thank you for your time.